In this video, we will discuss about the mechanism of the results that we have observed in the previous discussion, that whenever this FABP5 protein expressed on the cell surface, there are more activated T cells in the ovarian cancer or in the periphery of the ovarian cancers. So in order to find out the mechanisms why this things is happening, they started by using the string analysis of this FABP5 protein, that uh, where this FABP5 proteins interactions occurs within the cells, and they are able to find some of the candidates that interacts with this FABP5 and in the Homo sapiens, and this is about this different organism. So, and in order to check whether this proteins has any role towards this FABP5 protein expressions, what it did is they did the immunoprecipitant assay and then did the LCMS. And what they have find out is that out of those candidate proteins, they are able to see that this TAGLN2 has the highest expressions. This indicates that there is some interactions that occurs between TAGLN2 and FABP5, and the hypothesis that they did is that this TAGLN2 helps this FABP5 proteins to expressed on the cell surface, and it helps in the transport of the FABP5 proteins from the nucleus to the cell surface. And this is the hypothesis they made so far. And thereafter, what they did is they further validated this TAGLN2 expressions using the Western blot of the through the immunoprecipitant assay, where they can see that the expressions of this TAGLN2 is clearly seen whenever they immunoprecipitate FABP5 proteins. So, and these things they are shown in the panel D, that can be seen here. And once they confirm that these TALNG2 expressions are high whenever this FAB5 protein is expressed on the cell surface, and they wanted to see whether this is really the case. In order to check their hypothesis, what they use is they use this TAGLN2 knockout cells and the TAGLN2 wild type cells, and did the immunofluorescence staining of this TAGLN2. And what they have seen is that in the wild type cells, this TAGLN2 and this FABP5 proteins are expressing in a way that this FABP5 proteins are in the cell surface. Expressions of these FABP5 proteins are on the cell surface of the cells, that is, they are surrounding the DAPI signals, and these things are quantified in the panel F, the first figure, where we can see that this TA, LNG, LN2 and FABP5 co-localizations is 100% in the case of these wild type cells, whereas in the case of knockout cells, they do not see any co-localization of this TAG, LN2 and FABP5 proteins. And here in the immunofluorescence also we can see that the red signals are on the it looks like to be on the periphery of this DAPI. Whereas in the case of this TAGLN2 knockout cells, what they have find out is that there is more overlap of these FABP5 proteins and DAPI, indicating that these FABP5 proteins remains mostly in the inside the cells or in the nucleus, because this TALNG2 is absent in those cells, and the FABP5 proteins will not be transported on the cell surface. So let me again explain this panel F. What it shows: first panel is the overlap of this TALNG2 and FABP5 proteins. So this overlaps, since in the case where this TLNG are expressed on the cell surface, then this FABP5 proteins are also expressed on the cell surface. So their overlap seems close towards 100%. But in the case of knockout, since TALNG2 is not present, it is close to 0. But when they check this FABP5 and DAPI overlapping, they can see that the overlappings are higher in the case of knockout, indicating that most of these FABP5 proteins in this TAGLN2 knockout cells remains in the nucleus, whereas this is not the case for the wild type cells. And then they did the reverse mechanism, that is, in the knockout cells they inject this TAGLN2 mRNA, and what they find out is that once they inject TAGLN2 mRNA, there is an increase in the FABP5 expressions on the surface that they have measured using the flow cytometer compared to the control mRNA, and this indicates that there is FABP5 has a direct role towards this transport of this not FABP5, this TAGLN2 has a direct role towards transport of this FABP5 proteins on the cell surface. And these things are quantified on the bottom figure of this panel G, where we can see that the surface expression of this FABP5 are statistically significant compared to the control mRNA injection case. So once they made these relations, then what they did is they wanted to check this. They wanted to check the CD8 plus cells. They wanted to check the CD8 plus activated cells relationship with the expressions of this FABP5. So what they did is they take the wild type cells and FABP knockout cells and the naive CD8 plus in the naive CD8 plus T cells, that is in the new deactivated CD8 plus T cells. Some of them are wild type and some of them are FABP5 knockout, and they use these two control mRNA. First one is this oval albumin mRNA, and second one is TAGLN2 mRNA. And then they did this wanted to check whether the cells are able to take this lipid or not. So in the panel H, they are checking whether the lipid intake is also affected by this TAGLN2 or not, and TALNG2 and FABP5 knockout cells or not. And what they have find out is that in the case where the FABP wild type and control mRNA is used, they see that high amount of they see that there is an increase or high amount of there is an increase in the we can say there is an increase in the lipid uptake, which is they measured using this CD8 plus cells and CD44 T cells. And for the case where FAB5 is knocked down and there is a control mRNA is used, they see some decrease in the signals. And for the case where FAB5 knockdown and TAG-mRNA2 is used, they do not see, they see their signals is comparables to the or slightly lower or comparables to the case where FABP wild type and control mRNA is used. And in the case where they used FABP wild type and TAGLN2 mRNA, they see that more activated T cells are there. And these things are quantified on the right hand side, where we can clearly see that in the conditions where FABP5 wild type and TAGLN2 mRNA is used, they see that high amount of this CD8 plus T cells formations. And once they find out that the activated C cells are higher when there is a FABP5 is present and TAGLN2 are overexpressed, then they wanted to check with their lipid expressions. So when they want to check the lipid expressions, what they have find out in the lipid expressions is that they compare this palmitoleic acid and oleic acid, as these are the two lipids for that this FABP5 proteins controls. And what they have find out in the case of control, there is a decrease, there is some amount of this oleic acid formations and oleic acids intake and palmitoleic acids intake. However, in the case where TAGLN2 mRNA is used, they see the increase in the oleic acid and this palmitoleic acid intake compared to the all other conditions. So this indicates that the lipid intake is increases whenever there is a FABP5 is present, and also this TAGLN2 are expressed on the cell surface, and both of them, when both of them are expressed on the cell surface. And thereafter, and these things are we can see that quantified fatty acid content in the panel K, where we can see that for the multiple replicates there is an increase in the fatty acid content in the activated T cells in the case where wild type cells are used and TAGLN2 mRNAs are used. And this one, the left hand side of the panel K shows for the palmitoleic acid, and right hand side of the panel K shows for the oleic acids. So in both the cases, this for the wild type and TAGLN2 mRNAs conditions, there is an increase in the those lipid uptake. And after that, what they did is they check it in the cancerous conditions. So when they check it in the cancerous conditions, they focus mainly on this oleic acids, as these expression levels are higher and can easily be measured and can easily be compared between different conditions, measured or compared between different conditions. So, and these are in the panel L represents the major fatty acids that are found in these Ascites conditions in the ovarian cancer, and this is shown here that in the oleic acids are usually present in the 39.8%. And when they check this oleic acid relative abundance in different conditions, what they have find out is that when they use this control mRNA and Ascites, they see a decrease in the oleic acids relative abundance compared to the case where control mRNA is used and there is no Ascites. However, when they increase or when they use this TAGLN2 mRNA or overexpress TAGLN2 mRNA or overexpress TAGLN2 on the cell surface of this CD8+T cells, they see that increase in the oleic acids, indicates that in the cancerous conditions also this TAGLN2 helps the FABP5 proteins for lipid uptake, and these things are quantified in the panel N also, where we can see that in the conditions where wild type CD8+T cells and TAGLN2 mRNA is used, there is an increase in the fatty acid contents in the CD8+T cells. So this indicates that this TAGLN2 assists FABP5 in lipid uptake in activated T cells, normal conditions also, and also in the cancerous conditions or in the Ascites conditions in ovarian cancer. So I hope you understood how they conclude or how they did their study to validate to come up to this point. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.